Hello, my name is Joshua Mugabe. I welcome you to Enzymes. What are catalysts? Catalysts, these are substances that reduce the activation energy of a chemical reaction, facilitating it and making it energetically viable. A catalyst increases the speed of a chemical reaction. What amount of catalyst is consumed in the reaction it catalyzes? Catalysts are not consumed in the reaction they catalyze. Is there a difference between the initial and final energy levels in a catalyzed and non-catalyzed reactions? The catalysis does not alter the energetic state of reagents or products of a chemical reaction. Only the energy necessary for the reaction to occur, that is the activation energy, is altered. What are enzymes? What is the importance of enzymes for living beings? Enzymes, they are proteins that are catalysts of chemical reactions. From chemistry, it is known that catalysts are non-consumable substances that reduce the activation energy necessary for a chemical reaction to occur. Enzymes are highly specific to the reactions they catalyze. They are vital importance of life because most chemical reactions in cells and tissues are catalyzed by enzymes. Without enzymatic action, those reactions would not occur or would not happen in the required speed for the biological processes in which they participate. What is meant by substrate of enzymatic reactions? Substrates, these are reagent molecules upon which enzymes act. The enzyme has a special binding site for the attachment of its substrate. These sites are called activation center of the enzyme. Substrates bind to these centers, forming the enzyme substrate complex. What are the main theoretical models that try to explain the formation of the enzyme substrate complex? There are two main models that explain the formation of enzyme substrate complex. There is the lock and key model, and then the induced fit model. In the lock and key model, the enzyme has a region with a specific special conformation for binding the substrate. In induced fit model, the binding of the substrate induces the change in the spatial configuration of the enzyme for the substrate to fit. How does the formation of the enzyme substrate complex explains the reduction of the activation energy of chemical reactions? The enzyme possibly works as a test tube within which the reagents meet and they form products. With the facilitation of the meeting provided by the enzyme, it is easier for collision between the reagents to occur. Thus, the activation energy of a chemical reaction is reduced. This is one of the possible hypotheses. Of what structure level of the enzyme? Primarily, secondly, tertiarily, or quaternary? Does the enzyme substrate interaction depend? The substrate binds to the enzyme in the activation centers. These are specific three-dimensional sites. Thus, they depend on the protein tertiary and quaternary structures. The primary and secondary structure, however, condition the other structures, so they are equally important. What is the activation center of an enzyme? Is it the key or the lock of the lock-key model? The activation center is a region of the enzyme produced by its spatial conformation to which the substrate binds. In the lock and key model, the activation center is the lock and the substrate is the key. Why can it be said that enzymatic action is highly specific? Enzymatic action is highly specific because only specific substrate of one enzyme bind to the activation center of that enzyme. Each enzyme generally catalyzes only a specific chemical reaction. What happens to a denatured enzyme regarding its functionality? How can that result be explained with the help of the lock and key model? According to the lock and key model, the enzyme functionality depends entirely on the integrity of the activation site or center, a molecular region with specific special characteristics. After denaturation, the spatial conformation of the protein is modified. The activation center is destroyed, and the enzyme loses its catalytic activity. What are the main factors that alter the speed of enzymatic reactions? The main factors that change the speed of enzymatic reactions are temperature, pH, substrate concentration, that is the quantity. How does the substrate concentration affect the speed of enzymatic reactions? Initially, as the substrate concentration increases, the speed of the reaction increases. It happens because the free activation centers of the enzyme bind to free substrates. Once all the activation centers of the available enzyme become bound to their substrate, new increment of the substrate concentration will not affect or affect the speed of the reaction. How does temperature affect the action of the enzyme upon their substrate? There are defined temperature ranges under which enzymes operate. There is a specific temperature level, which we call the optimum temperature, in which the enzymes have maximum efficiency. Therefore, temperature variation will affect enzymatic activity and the speed of the reaction they catalyze. In addition, as proteins, enzymes can be denatured under extreme temperatures. Concerning enzymatic reaction, how different are the graphic curve of the variation of the speed of reaction as function of substrate concentration and the curve of variation of the speed of reaction as function of temperature? The curve of variation of speed of enzymatic reaction as function of a growing substrate concentration is a growing curve until the point where it stabilizes due to the saturation of the activation centers of the enzymes. The curve of variation of speed of enzymatic reaction as a function of growing temperature has a crescent portion and reaches a peak, which we call the optimum temperature. Then it decreases and it reaches zero, the point of inactive of the enzyme by denaturation. How is the cooling of organs and tissues for medical transplant associated with the effects of temperature upon enzymatic reactions? The molecular degradation during the decomposition of organs and tissue is catalyzed by enzymes. On cooling to adequate temperatures of some organs and tissue destined for transplantation reduces the enzyme activity and thus lessen the natural decomposition process. By the same rationale, cooling reduces the metabolic work of cells and prevents degradation of their own substructure to obtain energy. Elevation of temperature later reverts denaturation of enzymes, and the organs and tissue also preserved by other specific techniques may be grafted into the receptors. How does pH affect the enzyme activity? The concentration of hydrogen ions in solution affects enzymatic activity. Enzymes have maximum efficient under optimum pH. Since pH is one of the factors for the denaturation of proteins, if an enzyme is submitted to a pH level under which it's denatured, there will be no enzymatic activity. Do enzymes act better under acid or basic pH? Most enzymes, they act in a pH between 6 and 8, range that corresponds to the general acidic level of cells and blood. There are enzymes, however, that act only under very acid or very basic pH. So enzyme activity depends on pH interval. In the stomach, for example, gastric juice has a very low pH, around 2. There, the enzyme pepsin acts to intensely digest proteins. In the duodenum, the pancreatic secretion increases the pH of the enteric juice for the action of other digestive enzymes. For example, trypsin. Since pepsin, a gastric enzyme, does it have acid basic optimum pH? What happens to pepsin when it passes into the duodenum? Pepsin acts within the stomach, so its optimum pH is around 2, an acid pH. When the enzyme passes into the duodenum, it meets a higher pH and its enzyme activity ends. What are enzyme cofactors? Some enzymes, they need other associated molecules to work. These molecules are called enzyme cofactors. They can be, for example, organic ions like mineral salts or organic molecules. Inactive enzymes, which are not bound to their cofactors, are called apoenzymes. Active enzymes bound to their cofactors are called holoenzymes. What is the relationship between vitamins and enzyme cofactors? Many vitamins are enzyme cofactors that cannot be synthesized by the organism and must be obtained from the diet. For the enzymatic reaction, what is the effect of the substance with the same spatial conformation as an enzymatic substrate? How is this type of substance known? Substances that simulate substrates can bind to the activation center of enzymes, thus blocking the true substrate to bind to these enzymes and paralyzing the enzymatic reaction. Such fake substances are called enzyme inhibitors. The binding of enzyme inhibitor to enzyme can be reversible or irreversible. Many chemical drugs, for example, some antibiotics, viral, and neoplastics, and hypertensive, and even sildenafils, trademark Viagra. They are enzymatic inhibitors that block enzymatic activities. What is the action mechanism of the antibiotic penicillin. Penicillin was discovered by a Scottish doctor, Alexander Fleming, in 1928. This is a drug that inhibits enzymes necessary for the synthesis of peptidoglycans, a constituent of the bacterial cell wall. With the inhibition of the bacteria, population stops growing because there is no new cell wall formation. Fleming won a Nobel Prize in medicine for the discovery of penicillin. What is the action mechanism of the antiretroviral drug called protease inhibitors, which are used against HIV infection? Protease inhibitors are some of the antiretroviral drugs used to treat HIV infection. Protease is an enzyme necessary for assembling of HIV after the synthesis of its proteins within the host cell. Protease inhibitor binds to the activation center of the enzyme, blocking the formation of enzyme substrate complex and the enzyme activity, thus impairing the viral replication. What are allosteric enzymes? Allosteric enzymes are those that have more than one activation center to which other substances called allosteric regulator bind. Allosteric regulators can be allosteric inhibitors or allosteric activators. The interaction between allosteric enzyme and allosteric inhibitors disallow the binding of the substrate to the enzyme. The interaction between the allosteric enzyme and allosteric activator allows the binding of a substrate to the enzyme and sometimes increases the affinity of the enzyme for the substrate. This regulatory phenomenon of the enzymatic activity is called allosterism. What are zymogens? Zymogens or proenzymes, these are enzymes secreted in inactive form. Under certain conditions, zymogens shift to the active form of the enzyme. Zymogen secretion in general happens because the enzyme activity can harm the excretory tissues. For example, pepsinogen is secreted by the stomach and becomes active under acidic pH, turning into enzyme pepsin. Other well-known zymogens are trypsinogen and chymotrypsinogen enzymes that are secreted by the exocrine pancreas, in which become trypsin and chymotrypsin respectively. Thank you very much for participating in Enzymes. Please do like, share, and subscribe.